Yeah, well, hello everyone. How's everyone doing? As we can see, the quarantine has taken a toll on me, and I blew up Britain a couple of days ago. So, yeah, when I saw this initiative, I think it was great. I mean, we don't have anything better to do at the moment, so I decided to talk a bit about what I do. So I'm a postdoc at the Open University, and I am working on potential life on Mars and how to try to identify it. So I'm going to share my screen. So I have a short presentation, and afterwards we can just find me questions and discuss about it if you want. So, right, let's get on with this. So, my my main question to try to answer at the moment is trying to answer what David Bowie asked in the 80s, is there Mars? So humans have been fascinated with potential life on Mars for a very long time. So in 1897, Herbert Wells wrote The Wall of the Worlds. That was the first time that actually Martian life was depicted. But it was until 1911 when Burroughs wrote the Martian Tales, in which they describe Martians as these tiny green men. Then 1948, we have Chuck Jones, Marvin the Martian. 1951, we have The Martian Chronicles by Bradbury. Then 1973, no, the 80s, sorry, we have David Bowie asking the question about, is there life on Mars? And then 96, sorry, Tim Burton made one of my favorite Martian films, Mars Attacks. And then 2015, Ridley Scott and The Martian, which actually is quite an accurate scientifically speaking film. So when we talk about life on Mars, are we thinking more of Marvin the Martian, or is it going to be more like some kind of microbe? Well, it is more likely to be some kind of microbe in this short talk. So first of all, we need to ask ourselves, what are the requirements for life? So the first thing is, for life, we need to have the building blocks. That's what we call the chomps. That stands for carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Then obviously life thrives using energy. So we need to use some kind of energy source in order to survive. So for this, we tend to use reduced and oxidized substances in which the electrons are going to be taken from one substance and transferring to the next. We also need some radiation shelter, because obviously radiation can be quite obnoxious for life. We need an optimum temperature, and we need water. So the question is, in which kind of places in our neighborhood, to say the least, we have this? So obviously we have Mars, and we have the icy moons in Jupiter and Saturn, such as Enceladus or Europa. But my research is focused on Mars, and this is what I'm going to be talking about. So let's talk about Mars. One of the most important questions is there water on Mars, or is there water on Mars? Well, 4 billion years ago, apparently Mars was quite a wet planet. It had nice and thick atmosphere that allowed the presence of liquid water. At some point, we don't really know why, although there are several theories, such as the cooling down of the nucleus that led to the loss of magnetic protection for the atmosphere. The atmosphere of Mars started to be depleted, started to disappear, started to thin. This meant that a lot of the pressure that was on Mars disappeared, making the presence of liquid water very hard. And over time, the drying out of Mars led to the current situation we have nowadays, in which we tend to think of Mars like this very barren, this very dry and very reducing planet, oxidizing planet that we have nowadays. So, but the burning question is, how do we know that there was water on Mars? Well, the picture on the left is what it seems to be a delta. So deltas are associated to rivers, they're associated to running water. So it seems that water was present enough and for enough time to make some incisions on the surface of Mars, making this kind of geomorphological features that we can identify on Earth. On the right-hand side, we also have a sulphate deposit on Gale crater on Mars. We can think, why is a salt deposit important on Mars? Well, salts appear when there's interaction between water and rocks, and there's a dissolution of the rocks, and then when the water evaporates, these dissolved substances in the water are going to precipitate in the form of salts. So then there is further evidence that Mars had water in the past. But is the presence of salts in Mars also important for life? Well, guess what? Yes, for three reasons. First of all, the presence of salts in water, we all know that tends to depress the freezing temperature of the water. That means that the presence of salts in liquid water are going to make the solution harder to freeze. But also, on the other part of the scale, it is going to also make it harder for the water to evaporate. And this is particularly important on Mars, because if we think, I mentioned that Mars has a very thin atmosphere, which means that the pressure is going to be very low, which means that even at very low temperatures, the water on Mars can evaporate. And there is a very important third process called deliquescence, in which the presence of certain salts can scavenge water from the atmosphere and actually can make these salts to dissolve to generate a brine, a very concentrated salt concentration on the surface that can then percolate, or they can infiltrate into the Martian ground. So the very burning question that we have is, how do we try to identify life in other planets? Because obviously it is very hard for us to go there, but we can use certain proxies, certain substances, certain signals that we can detect from afar, and we can relate them to the presence of life. One of them, and it's very important, is methane. Methane on Earth is produced 90% by biological agents. They are microbes called archaea that are going to be very important in the rest of the talk. But what happens with the other 10%? The other 10% comes from another kind of reactions that are independent of life. One of them is geochemistry, so the reaction of water and CO2 and hydrogen can generate this methane. But also we can also have thermogenic produced methane, in which we have complex organic molecules at high temperatures, they can break, as happens when we are fractionation oil, and generate methane. So the important thing about this is that, yes, we have methane and we have other gases, but actually not all methane is the same. There are certain hints in the molecule that can tell us whether methane is coming from a biological or from an abiotical source. One of them is the molecular weight of the atomic weight of the methane, because the presence of certain atoms that are going to be lighter, carbon-12 compared to carbon-13, is going to be indicative of life. Because microbes tend to be on a diet. They prefer to go for lighter substances instead of more heavy substances. And also, it's very important, especially at the time of looking for life in other planets, to pay attention to the ratio between methane and other gases such as ethane. Because microbes tend to be very specific. They tend to produce huge amounts of methane and not other gases. Whereas if we have another kind of reaction that involves breaking, for example, the molecules of very heavy hydrocarbons, we're going to have a whole range of different gases, and there's not going to be an enrichment on methane in particular. So how can microbes produce methane? Well, there are three ways that microbes can produce methane. One of them is taking acetate. and breaking the molecule. Acetate has 2 carbons. So they break the molecule in the middle, generating on one hand methane, and on the other one is going to produce CO2. There is another method called hydrogenotrophic, in which they can make react CO2 and hydrogen in the cell to produce methane. But more recently, and this is particularly important there is a third method called methylotrophic methanogenesis, in which certain substance that contains methyl radicals in the structure can actually be broken by microbes to produce energy and produce a lot of methane. Why is this important? Well, one of the most controversial things or topics on Martian science is whether we have presence or not of methane. Because there have been several missions, both orbiters and rovers, that have identified methane on Mars, but also there have been some others that haven't. So there is this very agitated conversation between Martian scientists to whether proof or disprove the presence of methane. And actually, quite recently, Curiosity on Mars identified peaks of methane on Gale crater that then were confirmed by the Max Express orbiter. But then quite recently, we sent the ExoMars mission to Mars, which is specialized in the detection of methane, and it couldn't find methane. I'm not going to get into the detail why this could have happened, but if you want later on the questions times, we can talk about it. So we've talked about Gale crater, but why is Gale crater interesting from a scientific point of view? We have the MSL there, the Mars Space Laboratory, so that's the Curiosity rover, and it has identified methane there, which is great. But when we go back to Earth and we see the kind of environment that Gale crater could have been in the past, we kind of have a problem, because people tend to assume that the presence of sulfates inhibit the production of methane. It's important to mention that a very important part of Gale crater is covered on sulfates that indicate the presence of water, but also, as I just mentioned, what does this mean for methane production? Well, the first 2 routes of methane production by microbes, the one that uses CO2 and hydrogen and the one that breaks acetate, is inhibited by the presence of sulfate. But the third type of route that I've mentioned, the methylotrophic one, actually can happen on high sulfate concentrations. So we have this very rich in magnesium sulfate environment on Mars. So we need to take a step back and think, do we have any kind of environments like this on Earth? Guess what? We do have them. They are rare. I'm not going to lie, they are rare, but they are present in some places in the world. So we have some magnesium sulfate rich lakes in Spain, in Australia, in the States, and in Canada. And actually, the lakes I'm going to be talking about today are in British Columbia, on the western coast of Canada. The picture on the left is how the lake looks at the end of summer. So we can see that there's been a lot of evaporation. And if we see around the red rim, we have a lot of white spots. That is all salt. But actually, if we actually are able to put our hands under the mud that we have on the top of these ponds, we actually find a very, very, very thick layer of salt precipitated in the water. This means that the concentration of salt, of magnesium sulfate on this water is higher than the water can take, making it to precipitate. So it's kind of the kind of situation that could have happened in Gale crater when it was drying out. So there are 3 lakes in Canada that I base my research on. The first one is the Basque Lakes that have a very strong salinity. It's like 34% salinity. So 34 grams of magnesium sulfate per 100 grams of water. And we can see all these salt crusts around the lakes, around the ponds. Then we have Salt Lake, Clinton Lake, which is a bit less concentrated, but is kind of similar. And then we have a third lake called Last Chance Lake, which has a completely different salt, but is also kind of relevant to the kind of conditions that could have happened in Mars in the past. So this is me doing my sampling. So obviously I have to still carry my rainbow when I'm doing some sampling. That's what I do. And you can see that the kind of lake I work in is not the kind of lake that you would like to go for a swim in. They actually stink of sulfide because of microbial activity. They can actually break the sulfate molecule and produce some very nasty gases. And then the picture of the right, so the picture of the left was a glorious day, 20-something degrees, great. The picture after is the day after, we're talking about 10 degrees, it was raining, it was a terrible day, it was a terrible day. And what I'm doing on the picture on the right, if you see there is a tube on my hand, this is how I sample gases from the surface. What I have on the right-hand side of the picture is a gas sampler that makes a vacuum, and there is a bag inside, which actually absorbs the water, the gas on top of the lake, and stores it in these bags that then I can bring to the lab to analyze. So, great. We have these places, we have these gas samples, but do we have methane on these gases? Well, this can be kind of disappointing. So, on the baseline, this is the general amount of methane that you can find outside of the lakes. And we can see that Bass Lake and Last Chance Lake, there is no difference to them, to the baseline, except for this point up here, which actually, it was kind of a very lucky mistake, I could say. I'm not going to say mistake, but maybe a mishap. So one of my collaborators was actually taking some course on these ponds. And when she took the core out, I could see that there was gas coming out from the core. And I immediately ran like a crazy person to grab the sampler and go to the pond and put my sampler there and try to get some gas. And actually, we can find that, yeah, there is quite a significant amount of methane compared to the baseline. And BL1 is another of the Basque lakes, and we can really see that there is methane production. So it seems that there is methane production, but this methane is produced not in the water itself, but more in the sediment that is underneath the water. So this is how my bench looks at the moment. So this is sediment that we've taken from those lakes, and we mix them with a medium in the lab, try to enrich the cultures and to see if we can get any for gas production. So for that, we add different substrate, as I mentioned before, acetate, SO2, methylated amine. So this third kind of reagent, this third kind of substance to see if we can have any methane. And what we can find, so this is one of my cultures after several weeks of incubation. You can see that there are some bubbles in the sediment. If we have a close-up, we can really see these bubbles here. So then what we can do is extracting the gases from the surface of the bottle, so around here. And actually, we can see that when we have trimethylamine, this extra substance that is going to be the leading substance on the path, we get to produce up to 15% of methane on the headspace. That is a heck of a lot of methane, especially regarding that supposedly the kind of conditions that we are inhibiting the production of methane. So what does this mean? Well, we are going to have these archaea in the sediment that are going to be able to use trimethylamine, but not acetate SO2, because all the bacteria called sulfate-reducing bacteria are going to scavenge those reagents and use them for themselves, but they can't use trimethylamine. And this trimethylamine is going to feed our archaea. But the question is, what is this trimethylamine? Because it sounds like a very complex molecule coming from. Well, actually, what we found is that some of the microbes that are surviving in this kind of environment that is very saline, very strong for them, to protect themselves, they produce certain substances that are going to prevent the salts from entering into the cells and breaking them. One of them is called choline. And choline, in the presence of certain microbes, can be broken into trimethylamine that then is going to be feeding archaea that are going to be producing. So why is this? Obviously, I mentioned that this kind of environment is going to be similar to how Mars looked when it was drying out. But also, one of the things that my research group is very good at doing is we have people in the group that are specialized in simulating, in modeling how water in Mars could have been in the past or how it could be nowadays. So one of the things that we've done is using these simulated brines, and we've taken some of the microbes that we've grown from the lakes and transfer them into the brines to see if they can survive in these conditions without having to add anything extra. So what I'm showing in this picture is actually the presence of a substance called ATP that is a proxy for life. The more proxy that there is in the medium, the more microbes are going to be there. And actually, we can see that some of them is very low, it's almost zero, but some of them can be quite high compared to the baseline, indicating that actually microbes can survive on these conditions. But then how does this link to the main question that I have is, can we detect methane on these Martian brines? Guess what? We can actually find some methane on these brines. And if we look at the legend at the bottom, it's actually trimethylamine what is actually helping to produce this methane, although we haven't added it on this medium. So it seems that there are some of the microbes that are able to produce this trimethylamine that is going to be feeding our archaea. So I think that I am going to leave it for now. And my take-home messages for today is, well, Mars could have had conditions in which life could thrive in the past. Methane can be produced in sulfate-rich environments using methylated amines, and life can thrive in modern Martian brines and produce methane. So if we want to ask any questions, please be ahead.